all the delegates. A very good morning to all of you. First of all, I would like to thank CDI to provide me this opportunity. Today I am going to present my work on development of diabetes, development of multiple reaction monitoring assay for quantification of carnosine in human plasma. And this work is essentially in collaboration with Dr. Undi Krishna from CDI. So there might be three questions in your mind. Why it is important to quantify carnosine in human plasma? Why MRM method? And what is its relevance to the diabetes? So to answer this question, I would like to introduce two important players, carnosine and carnosinase. So all the delegates present in this hall, pending hall, we have these molecules in our body. A small molecule, carnosine, which is a dipeptide, and the enzyme carnosinase, CN1 and CN2. CN1 is present in the plasma and CN2 in the cell. So, what is the importance of carnosine? Carnosine is a powerful antioxidant. It involved in glucose metabolism. It is a neuromodulator, and it protects biomolecules like protein and DNA. Now, during my PhD from IMTech Chandigarh, I worked on the first part, that is carnosinase enzyme, and find out how the substrate recognition is actually occurs. And we saw the first crystal structure of human CN2. As you can see here, it is composed of two chains. This is the catalytic site, and I would like to tell that CN1 is more active on carnosine, CN2 is less active. But what was the reason? It was not known. We found out the molecular features that in CN1, serine active site is protruding inside the active site, and it is replaced by glycine in the CN2. Now come to the point why carnosine and carnosinase are important for the diabetic perspective. So these two slides will make you agree that yes, carnosine and carnosinase are really important in terms of diabetes and diabetic nephropathy. In 2005, Jensen et al. Have shown that carnosine is a protective factor in diabetes nephropathy, and the enzyme CN1 is associated with development of diabetic nephropathy. How? They have done studies on European populations, and what they found in the signal peptide sequence of the carnosinase, there are 5 leucines and 6 or 7 leucines variants are also there. Those persons which have 5 leucines, they are resistant to diabetic nephropathy. There is less chance of diabetic nephropathy. But if they have 6 or 7 leucine repeats, they are susceptible to the diabetic nephropathy. Why? The molecular reason is if more leucines are there, the secretion of the enzyme is more, then the carnosine level will go down because it is a substrate, and the protective effect will go away. I would like to introduce another important study by the nephrology and endocrinology department of PGI Chandigarh. On North Indian population, they have studied and found that these variants is actually present in Indian population also, and they found that if 5 leucines are there, they are resistant towards the diabetic nephropathy. Further, it has been shown that carnosine influences glucose metabolism, and this study shows that progression of diabetic nephropathy to ESRD, end-stage renal disease, is associated with the polymorphism in the CNDB1. The next slide. Carnosinase attenuates the development of diabetes to diabetic nephropathy, and in a very prestigious journal of JCI, it has been shown that carnosine mitigates the obesity disorders by reducing the carbonized stress. So, what is the take-home message right now? That in diabetic nephropathic patients, the carnosinase activity is high, carnosine level is low, and they are more susceptible to the nephropathy. So, if one can monitor the carnosine level in the plasma, we can use as a biomarker. And to answer this question, we developed a novel method of targeted multiple reaction monitoring using the mass spectrometry. We use triple quadruple mass spectrometer, so there are 3 parts. We know that there are thousands and thousands of molecules present in the plasma, and to determine one particular molecule is really very we select a particular fragment of the carnosine. Here we selected 110 and 156. Now what will happen? This method selectively identify the carnosine and quantify. So this is the logic of our complete experiment. And with this what we did, we first prepared the standard curve using the pure compound, and then we performed the extraction and quantification of the carnosine. Blood plasma sample has been taken, 100 microliter of plasma. We precipitate the protein using the chilled methanol, speed wave drive, and run the whole method. Now what we actually found, this is the first time we found that carnosine exists in three forms: 227, 210, and 249. 227 is the actual MIC, 210 is the deaminated form, and 249 is the sodium adder, which generally found in the mass spectrometer. This panel shows 6 plasma samples from the healthy individuals, and the retention time has been shown, which shows the method is really reliable because the consistency of their retention time. All this work we recently published in a reputed journal of Royal Society of Chemistry, and this is the first method on the plasma carnosine quantification. Now what we are witnessing here in this is the first study where we have checked the carnosine level in 10 healthy individuals and 10 diabetic individuals, and what we found in healthy, the carnosine level, plasma level is 140 nanomolar, while in diabetic it is less than 80 nanomolar. So what is our observation is suggesting that right now what we are planning to do, we are going to check the level of carnosine in healthy, pre-diabetic, diabetic and diabetic nephropathy patients and determine their carnosine level, so we can use it as a potential biomarker. We extended our studies for the activity studies of the carnosinase. We purified both the enzyme after genetic engineering and cloning of both the enzymes, and we checked the activity and we found that our method is reliable in compared to control. CN1 hydrolyzes efficiently, and it is threefold more active than the CN2. So this is completely about my recently developed methods. And one another things which we are currently doing, that as we know that if we give the carnosine treatment to the diabetic patients or diabetic nephropathy patients, this molecule will readily hydrolyze by the enzymes. So that cannot be a good strategy. So what right now we are planning to do, we have derived certain carnosine analogs which may be resistant to the carnosinase enzyme. How? Because their structure is different. So we performed the docking studies and we found they indeed bind to the enzyme, and right now we are performing in vitro and in vivo studies to check if they block the enzyme, we can use them as a therapeutic approach. So with this, I would like to conclude, and I would like to acknowledge my mentor Dr. Mahesh J. Kulkarni, Dr. Unni Krishnan, Director NCL, and Sir for the fellowship. Thank you everyone, and I will be happy to answer questions. Thank you.
It's a very nice study. I would like to know, have you checked this carnosine and carnosinase level in the diabetic nephropathy patient? Because whatever you all showed is your published article, already published article. But what have you done on diabetic patients or related to diabetes? I know the level is less in diabetic nephropathy patient, but have you actually seen these values?
Yeah, this is a very good question actually, and I would like to answer in terms of both the molecule, carnosine and carnosinase. So carnosine we have checked in the healthy and diabetic individuals. First of all, this is the first method. There was no any previous reports on the checking method for the carnosine detection. Okay. Second thing is, right now we are doing the carnosinase level estimation in the plasma because CN1 is present in the serum. So we want to check the level of the CN1 in healthy and diabetic patients, but the method is not available. So what we are doing, we are using mass spectrometry. There is a method called parallel reaction monitoring. With this,
okay, fine. That you already you have developed the method, but have you seen levels at because you haven't shown levels in diabetic nephropathy patient or diabetic patient or control group?
Yeah, in diabetic we have done for the carnosine. For carnosinase we have not done because we are still developing the method for the carnosinase. Carnosine method is available. For carnosinase the method is not available. So that's why we didn't check in the diabetic or diabetic nephropathy patients. But for carnosine we are doing for the diabetes as well as the diabetic nephropathy.
So So can this level be used as a kind of to see patients who are more susceptible to diabetic nephropathy or as a prognostic indicator?
Yes, definitely. We believe on this. We are working on this hypothesis, and we expect that in nephropathy, because the enzyme activity is high, the level of the carnosine must be low, and we can use it as a biomarker for the early prediction of the diabetic